Yes.
Awesome. So, hi everyone, welcome to our High-Affinity Talks. This is where we give researchers the spotlight to present their new antibody-related research. We're really excited for today's topic, which is all about the affinity of B cells following a COVID vaccination. A little bit about us, the hosts. I am Monica. I'm the head of go-to-market at My Laboratories, and we're also joined by Stan Posofsky, who is the co-founder and CEO. Just a quick introduction to who we are. Most of you may know us as the creators of MyXCR, which is the gold standard software tool used by thousands of researchers, cited thousands of times for immune repertoire profiling. We are also a software company, so we also have Platforma, which is our end-to-end biologics discovery solution. Just for you all to know, our software has been cited thousands of times. It's used in 8 out of the 10 biopharma companies and also used by all of the top 50 research institutions around the world. So we are quite proud of this. Today's topic is led by Thomas Dupic, who is the team lead at the Center of Immunology in Marseille. And he'll be talking about the affinity and evolution of B cells following a COVID vaccination. He will be presenting his topic following a longitudinal study of 10 individuals post COVID infection / vaccination and talking about how the lineages expand, what separates people who are responders from non-responders, and also how he can identify reactive clones. So for today's agenda, we'll have Toma presenting his slides and his topic for around 25 minutes, and then he will be joined by Stan in the discussion portion. So for that, feel free to drop your questions in the chat or in the Q&A, and we can address them at the Q&A section. Okay, Toma, I will pass it over to you.
Thank you, Monica. So, as said, I'm currently a researcher at CIML in Marseille, and I'm going to talk about a project that I did during my postdoc, so the last postdoc project in the lab of Michael Desai at Harvard with Alif Moulana. And this project is about the dynamic of the spike-specific B cell repertoire, but particularly on what we can do to detect affinity and measure affinity in a BCR repertoire. So I'm interested, like I think many among you, I'm interested in the B cell repertoire. So this B cell repertoire is the set of all different BCR present in an individual on B cells, and it's extremely high diversity with at least 10 to the 9 different receptors in one individual. That's more than all the other proteins put together. So it's extremely high diversity. But these BCR genes are very easily accessed. We can sequence them easily, both in bulk and single-cell RNA experiment. And the amount of BCR data available, like in the world, has been increasing exponentially since people have started doing that. People have started sequencing BCR. In particular, even when people are not looking directly at BCR receptor for their property as antibody, they're often using it as like a barcode to tag cells in many different contexts. So even that data is useful in any way. But the BCR repertoire in itself has a very huge potential. It can be used to do prognosis, right? You can use the BCR present in one individual to try to predict how is its disease going on. You can do diagnostic and medical history in theory, because while antibodies tend to disappear from the blood fairly quickly, the amount of memory B cells stay basically forever. So in theory, you can look at the memory B cell of one individual and see what has happened to him. And of course, you can use this BCR repertoire to look for potentially very good binder, potentially therapeutic antibodies. And all of these different points, I mean, people have tried to do things or have succeeded in looking at these things. So for example, for prognosis, for prognosis, I don't know of anything using BCR directly, but TCR have been used in the context of cancer, so the equivalent for T cells. For diagnosis, people have started using B cell repertoire to predict the disease of an individual patient. And for therapeutic antibodies, there's many examples of therapeutic antibody that come from BCR screens. But in general, I would say, given that the technology is 10 years old, the results have been a bit slow to come with. Like we have a lot of data, and the data is obviously very rich, but it has been hard to transform that into something that can be used effectively. And I think there's two reasons for that. The first one, and that I think a lot of people here are very aware of, is that PCR repertoire are hard to study. BCRs are randomly generated by VDG recombination. That means that unlike, say, all the other genes, we all have a different set of BCR repertoire. So it makes comparison between individuals very hard to do. It's even worse than that because BCRs of interest tend to mutate. They undergo affinity maturation, they go into germinal centers, they acquire mutation. And so they can create these sort of large clonal lineages that are formed of very many different clones, increasing the diversity and the complexity of the system. So all of these together, each individual repertoire is unique, meaning that most BCR are not shared, but not all of them are as likely as each other, and that you need to deal with all these complicated bioinformatic methods like clonal reconstruction, like phylogeny, means that BCR repertoire are hard to analyze. But I don't think this is the main issue with BCR repertoire. The main issue is that BCR are protein, are random protein, and in general, we don't know what they do. I mean, of course, we know what they are supposed to do. They're supposed to bind antigen, but we don't know which antigen. And BCR antigen interaction are very hard to predict. So current machine learning methods are not very efficient for prediction. I mean, I would say not at all, honestly, for antibodies antigen. And any sort of form of measurement is generally slow and expensive, specifically like the sort of gold standard that is an ELISA test, means that you need to express your antibody in a cell, you need to purify it, you need to test it against the antigen that you need to apply, but it's slow. So what I was doing during my postdoc and that I've been starting to do in my new lab is to try to develop other methods to characterize functional antibodies and to study the BCR repertoire. And to do this, we use yeast display. So we use antibodies presented in the scAV format that are expressed at the surface of B cells. So one type of antibody for each yeast, but each yeast can express a lot of different antibodies. And we mix this yeast with fluorescent antigen that we are going to detect through flow cytometry. Specifically, we're using a method that has been developed in the 2020s, which is called TIDE-seq. And TIDE-seq rely on the fact that each yeast can express a lot of antibodies. So we can take all of different sort of yeast, put them through a flow cytometer, and then sort the yeast depending on how many of these antibodies have an antigen bound to them. So basically here the blue antibodies have like way less in average, way less antibody than the light blue antibodies. And this means that we can estimate for each of these antibodies the proportion of antigen bound to them. We do this for a lot of different antigen concentration, and that allows us to create a full titration curve, like this, for as many antibody as we can put through a flow cytometer, basically. And this means that we can do library of size easily 10,000. We did at best 60,000 antibodies in parallel in one experiment that took, I think, 7 days. These methods work fairly well. They are compatible with titration. They are also compatible with the sort of other gold standard method that is biolayer interferometry to measure the KD. And we get a quantitative result. We get a KD, real value of like the affinity value of the antibodies. So we have used these methods in many different contexts, both for antibodies and for antigen. But in general, we have only used the method on one clonal lineage. So we never looked at a full repertoire because it's easier to look at, it's easier to make a library that has like a lot of different mutation on a given background, on a given antibody, than to look at real responses that are multiclonal and that involve a lot of different genes and a lot of different sequences. But with this project, we wanted to study them because this multiclonality is what is interesting about the repertoire. It's the fact that it can attack an antigen with very different type of proteins. So we wanted to know how many lineages are responding to the antigen. We wanted to know their properties. And we also wanted to know if, given this property, would we be able to detect them computationally? So will we be able to work without using this whole technology? So to do this, we needed a model, and this model is vaccination. So vaccination has been used a long time to study the repertoire response. I mean, it's great. You know the antigens, you know when the antigen were administered. So you know a lot, and a lot of papers have been using this to study the BCR. And in particular, we're looking at SARS-CoV-2 vaccination. So SARS-CoV-2 has been extremely well studied, and that's great for us because we know what's happening with it. We know that there's a short-term response in the first 6 weeks that's going to pump out a lot of plasmablasts, put a lot of antibody in the system, and then a more long-term response involving memory B cells. And we know, in average, how many of the B cells present in the blood, many of the PBMC B cells, are going to be positive to the antigen. And this number seems to be around 0.1 to 1% to 1% of the repertoire, 1% being at the peak of the response, so in the short-term window. So what we wanted to do was to follow this evolution of the BCR repertoire alongside SARS-CoV-2 vaccination. So we needed longitudinal samples, and we wanted to study specifically the long-term response. So looking at the early response, this big plasmablast response, is very nice, but it's also a bit cheating because in the case of vaccination, you're really interested in what happened in the long term. That's what matters. The sort of huge peak at the beginning is really not really wanted. So we wanted to look at the long-term response, the memory B cell response. We wanted to do it at high depth because we wanted to, have big phylogenetic tree, have huge clonal lineages, be able to follow clonal lineages between time point very effectively. And mostly we wanted to do functional characterization, meaning that we wanted to know what our BCR, or at least what a fraction of our BCR were binding to. So we collaborated with the Reagan Institute, the ULab at the Reagan Institute, and then they gave us a lot of samples, so longitudinal samples for 10 patients, and for a total of 10 to the 9 B cells. So that's a lot of blood. That's multiple liters of blood, I counted at some point. But that's what we needed to have this type of depth. We did a deep bulk repertoire sequencing, so heavy and light chain separately. So we didn't do single cell because that was way too many sequences to do single cells. 34 samples in total, each with 5 replicates, a lot of unique sequences. And then we analyzed them using MyXCR, of course. For clonal reconstruction, we used a software called Hilary, developed by Nathaniel Spisak, which is a bit better than the standard way of measuring clonal lineages. We used Igor to regenerate a recombination model. And for tree reconstruction, we used one of my software, which I call econometry and is not published yet, but is basically, I think, a better way to deal with large tree lineages. And so our immune repertoire looked like an immune repertoire. There was no real surprises. So if you're aware of how human repertoire change, they tend to vary a lot, meaning that you're never going to find the same B cells. You may not find the same B cells at different time points. That you're going to get a lot of new ones. But the statistical properties of a repertoire don't really change. In fact, if you look at this VGN frequency, for the different individual, and you look at that on a UMAP, they look very close to each other. And if you look at the different time point, I mean, sometimes they vary a bit, but most of the time they stay very similar to each other. So the statistical property of the immune repertoire don't really change. So that means that we don't, at least just looking at this like this, you don't see immediately antibody that you want to test, right? It's not like there would be a peak in the CDR3 length that show you, oh, this antibody is really reacting, we need to look at it. So when we had to decide which antibody are we going to look at to express that functionally, we didn't think too much. We just took the largest clonal lineage. So we took the 10,000 largest clonal lineages and then reconstructed, which is still a very small part of the overall repertoire, right? 10,000 largest clonal lineages only represent 0.1% of the total repertoire. There's a lot of small lineages. But we focused on these 10,000 largest clonal lineages, and we made a full library of their naive sequence. So for each of the clonal lineages, we reconstructed the naive sequence and then made a library out of it. So we had a big limitation because of our bulk sequencing approach. Again, we did bulk sequencing so we could have depth, but this bulk sequencing meant that we couldn't pair the heavy and light chain. So that means that to make the antibody, we had to have a combinatorial approach. We had to test all of the heavy chain that we wanted tested against all of the light chain. So to do this in practice, we bought oligopools from Twist, 10,000 variant of the heavy chain CDR3 and 1,000 variant of the light chain CDR3. We used templated VH region, so we focused on 10 different VH genes and 10 different VL genes. And then we created this naive library by just using golden assembly and created a very large library of 10 to the 7 sequence that we then sorted. So the sort went like this. We used the spike RBD protein. We put that on yeast. We sorted them first to check for expression, to check that the scAV was made. And we enriched them for binder, meaning that we selected things that looked like they were binding. We were not very specific during that time. We used the barcode to simplify sequencing, and then we used this tight-seq technology that I presented before to do this KD measurement. The final library size, so containing all of the sequences that we targeted as potential binder, contained 10,000 different antibodies. The advantage of using tight-seq and not just using enrichment, like if you're bit in that sector, lots of teams are using mostly enrichment, is that we really can differentiate cleanly binders from non-binders. So here we used this A value, which is like the top of the titration curve, to distinguish between the non-binder in our tight-seq from the binders. And we have even though we have enriched for binders, we see that we still have a lot of sequence that we characterize as binder. Similarly, and the sort of crazy result here is that it worked. Okay, that's and it works better than what we expected. Instead of having the sort of 0.1 to 1% numbers of B cells that were binding, we find that 2% of the heavy chain that we selected were actually binders. So 2% of all the heavy chain could be binding. And for the light chain, we find that nearly 25% of them were part of a heavy chain binder. So could be coupled with an heavy chain to be a binder. And we are able to measure the affinity, of all of these binders. And this is the distribution. Okay, so what are the particularities of this binding response of this clonal lineage whose naive sequence is a binder? So these lineage are very diverse. So we find all of the VGene we tested had at least a small population of binders. So there's no like big VGene shift, even though some of them are more represented than others. They also have very diverse CDR3 length. But they have properties that differ from the rest of the repertoire. In particular, they are larger. So the binding in orange are in general larger than the green one, which are just the one that express. They're also more likely to be public among vaccinated individuals. So they're more likely to be found in two or more of our different individuals, as you can see there. They also tend to evolve more. So we have multiple time points. So we can look at the lineage and how many new sequences appear in the clonal lineage compared to the number of sequences that are lost in these clonal lineages. And we see that this number increases for the binder compared to the selected sequences. We also see, without surprise, that they're more likely to be class switched than the express and the general sequences, are more likely to fall into IgG or AGA isotype than AGM and IgD. And I think the most surprising part for us is that they tend to evolve a bit differently. So here we're going to look at the evolution within a lineage, and we can look at a typical evolutionary metric, which is called the site frequency spectrum. And if you look at metric extracted from the site frequency spectrum, you see that clonal lineage are small, with a small difference, but a statistically significant one, are not quite the same as just selected sequences. So they tend to have a different evolutionary profile, which is probably not very useful to distinguishing binder from non-binder, but is very interesting from a theoretical and biological point of view. By contrast, if you look at the different isotypes, for example, you don't see this difference. So this is very much specific to the binders. outside of just the binding and either binder or non-binder, we find that the affinity of this germline naive original sequence also matters. High affinity germline, so this is among all the binder, high affinity germline are found across more time points, and high affinity germline, so still across the binder, are more shared across individuals. So germline don't just need to bind, they also need to bind well in order to be selected and to evolve. Before finishing, some rapid things on structural properties. From this experiment, because we're randomly associating heavy chain and light chain, we can also look at the importance of a light chain for specificity. And we see here that the light chain is very important for specificity, meaning you need to have a right light chain as an heavy chain. What you're seeing here is the number of unique light chain for a given heavy chain for the express sequences, which can have up to 10 different light chain, up to 100 different light chain, and for the sequences that are binder, which are most of the time associated with only one light chain. And this is even worse if you're looking not at specific light chain with a specific CDR3, but at the full V chain. Something else that may be interesting for this audience is that obviously we tried what machine learning could do for us. And on this specific data set, AlphaFold is not doing very well. So what you're seeing here is each dot is a specific antibody, and the circled in red sequences are the sequences for which AlphaFold told us that maybe our sequences were binders. So it doesn't work very well, but the sort of UMAP representation that you see here is obtained from AlphaFold. So it's obtained from AlphaFold internal representation. And so you're seeing that these sequences are more or less clustered. So we see that similar sequences are more likely to be binder, and that's not something that we could have seen without looking at like a machine learning representation. So I think there is still promise in these machine learning tools, but they need data, and that's what we try to give. So rapid summary, we sequenced 10 to the 9 B cells. We measured the affinity of 10 to the 4 antibody against a specific antigen, so the RBD of the spike proteins. We have characterized of this lineage that reactive antigen have differed from the non-reactive lineages. And yeah, repertoire are very interesting. You should sequence them. And yeah, I'd be very interested in looking at your BCR if you sequence them. What we plan to do in my lab in the future, obviously, we want to look at more antigen, want to look at more targeted data set. In particular, I want to look at single cell data set. We have more information about the specific cells and about the specific cell type. And we want to look at other parameters of the interaction. So in particular, the K-off is very interesting because it could be easier to get and easier to get with high precision than the binding affinity KD and the resistance under forces. And with that, I just need to mention that my lab is currently recruiting up to 2, but probably just 1 team leader. It's a great place. It's next to the sea. It's a very good immunology facility. So if you're interested, don't hesitate to apply. The deadline is fairly soon, May 1st. And I want to thank all the people from the Desai lab. So in particular, Alif, that I mentioned, did a lot of the job. So Alex Poré and Joao, Chao. And I'd like to thank my lab, of course, the TD lab, with Virginie and Charlotte. And with that, I'm happy to listen to question or discussion. Thanks a lot.
Thank you so much, Emma. So super interesting here. I will let San start with his questions, but feel free if anyone in the audience has any, to just submit it in the Q&A section.
Yeah, well, thank you very much, Thomas. That's extremely interesting. So much blood sequence B cells. So it's super, super interesting. And I have actually plenty of questions that I want to ask. But like while people are writing questions in the QA session, let me start with the high level one. So the first question that I have, since like people who are doing antibody discovery in the industry, we always look for like some ways to select best B cells with the highest affinity at the lowest price. Yeah. So there are a couple of questions here. The first one of the amazing properties that I see in your data set, that you demonstrated that there are distinct lineages with different V genes that converge to the same CTR3 motif. Yeah. So do you think this can be used as a predictor for the high affinity? I mean, if we'll, let's say, sequence on the one time point Then we look on the lineages and we're trying to find what CDR motifs are have more like diversity in the regions. Can it be used as a predictor of the affinity, and to what extent?
It's a good question. I think yes. So to answer very quickly, yes, it can be used as a predictor of affinity. We show that affinity that, for example, larger clonal lineages that are more sequenced tend to have a higher affinity. So, and that's true, and that has reason to be true across clonal lineages too. So that so that you could use similarity between CDR3 to sort of select for high affinity binder. I think the question of high affinity is also, I mean, the question of high affinity for industry is also a complicated question. And what exactly affinity mean? I think the gold standard that people measure, which is affinity at equilibrium, may not be the best representation of what is effective, what a good antibody is. And so, yeah, I think there's basically two questions. The question, which is what should we look at? And is that affinity at equilibrium, which is what we're looking at there? And can we use BCR repertoire data to find out good antibodies? Yes, but right now I would say not just this. You do need to have like some way, not just to check, but to sort of screen your antibody at this point in time. If that answers the question.
Yeah, well, thank you very much. And the related question here. So when I was working in the academia research, B cell repertoire is like you have to sequence a lot, and the deeper the better. But today, especially with single cell technologies, and when we go to the industry, we need, like for in vivo discovery, we need heavy light pairing, yeah, so ideally natural one. So more and more people use single cell, and single cell becoming more democratized, cheaper, still very expensive. But do you think, or let's put it this way, what level of depth of sequencing we need to be able to get some confidence in our antibody selection? And can modern single cell technologies offer that level of required depth?
Yeah, I think yes. I think okay. For there's two things, right? There's if you're looking at PBMC sequencing, which is what we're doing here, you're basically in the worst case scenario. Like blood PBMC in human is well known for being extremely diverse, for having a lot of naive sequences, and so you need to basically sequence way more than in any other situation. But even though we just here in our approach, we just look at the largest lineages, like very much the largest clonal lineages. That's a very small part of our repertoire. That's just 0.1% of what we sequence. And so we could have decreased the depth by a factor of 1,000, which would have put us right in the single cell sequencing range and still have had a lot of sequences. But then I would add that when people looked at single cell in like tissue, for example, or single cell in mice, you're not in the same situation at all. You're seeing things that are reacting to something most of the time, right? It's not like PBMC in humans. There's way less diversity.
And when you say, that's one of my questions, when you say you pick the largest lineages, largest by what? By the size in terms of the distinct clonotypes, or by the abundance?
Abundance in terms of RNA reads, which is a bit of a biased measure because it tends to favor plasmablast and the like, but still, it's abundance by number of reads. So that's our proxy for number of cells, basically.
Okay, and since you mentioned mice, so that's one of my questions. So this approach was human species, yeah? But do you think the same will be observed in non-human species? Same patterns. I mean, immune system should work the same, but still.
Yeah, the system works the same. We do have library made of antibodies from mice. I think I mean, you wouldn't look at the PBMC repertoire in mice because mice don't have a ton of blood, and so it's generally not very interesting. When you're looking at lymph node, typically you would expect to see many more binders than what we are seeing here. So you would expect to see the proportion of binder to be more important, and so you would expect them to be easier to detect in that scenario.
Thank you very much. I actually have a lot of questions, and now let me just go through a couple of technical ones that really matter for me. So the lineage reconstruction and in general the annotation of the antibodies. How important do you think the accurate annotation of the antibodies, the clonotyping, error correction, let's say including unique molecular identifiers in the sequencing to be able to reconstruct like the to correct for the sequencing and PCR errors? And what are the methods in your experience work best? I mean, you showed that you use Hillary and yeah, but like how important it is? Can we just cluster them by CDR3 and and that's it?
So it depends what you're looking at, really. I mean, if you're in academy, you will probably be looking at you can have different interests. But if you're interested in clonal lineage evolution, typically, as we were when we worked on this, having exact clonal lineages was very important, because if you don't have exact clonal lineages, typically you're going to take some of your clone are going to be sort of weird aggregate of things that have never seen each other, that have never evolved together. And so you create a big bias. And I think this bias has been seen in many papers before. That's something that I'm particularly irritated about. So I have a lot to say about it. But if you're looking at clonal lineages and clonal reconstruction, just to know if they are like attaching to something or if they have the same target, then you don't really care about this sort of accurate clonal ID. And then a lot of the typical approach that I use, typically using 10% similarity or that kind of things, they work very well. And I don't have any issue. In terms of is error rate important for us, I think it has become better and better with time. But particularly for BCR, where we're sequencing a fairly long region, right, with full V gene, and where we have mutation, and the random mutation random mutation are a problem. I would say they're not really a problem anymore for TCR, but for BCR, having UMI is important. And I think honestly, we could have done a better job with the way we did the sequencing. So we used multiple UMI, but maybe not at the right depth and like many different issues. But well, some of these issues can be solved more or less using cleaning up the data later on, but always complicated.
And related question, two questions. One question, so like when you use MyXR, for example, or other tools, it like alignment-based tools, it aligns against the germline reference set of genes. But there are allelic variants, and each individual has a allelic variants. So how important to distinguish the allelic variants against like somatic hypermutations? And what do you use here? Because historically, so we did a lot of such analysis, and like it may be important because like you think this is a mutation, but then like it's not. And the second part of the question, did you perform any benchmarks of the Hillary and method that you use for the lineage reconstructions against IgG5-ML, for example? That's I mean, I'm asking because many people asked us about this.
So for the first thing, yes, knowing the right gene is very important. Honestly, I just use MyXR for that, so that's all you. So there's the find allele function in MyXR that does that, and in our case, because we have a huge depth, we don't have any. I mean, it's easy to actually estimate which are the right gene and everything. So we have the right gene for this project at least. I think it's important, but I mean, it depends what you're looking at always. For the Hillary software, so I'm on the Hillary paper, so I need to mention that. So I do have a bit of a bias. I think the author check his methods against the different existing methods, and will tell you that his method is the best. I think at least on paper, it's the one most likely to give the right result at the end. I find that other methods tend to perform fairly badly on short CDR3. Which again for us is important. But again, lineage reconstruction, exact reconstruction is a bit of a niche problem, I would say.
For sure. And you need to have a really good depth to be able to reconstruct it. The last question I have about Hillary, because I also tested this myself and I actually run some analysis with it and I liked it and I recommend people using it. But will it work with non-human species same nice way as it works with human?
Good question. I think it's supposed to. That said, if you're starting to look at weird species, given that the affinity recombination, I mean, the affinity maturation mechanism become different, I wouldn't be so sure. So I think for mice, it's fine, but I wouldn't be pushing it to say chicken.
Sure. So let's other people ask questions. Yeah, Monica.
Great. Thank you, Stan. So we have a question from Valeria in the audience. She says, "Great presentation. Thank you. Looking at the SFS plots, I was wondering how you were controlling that these highly abundant SHMs are not allelic polymorphisms. Could you comment on this?"
Yes. So again, we group all our sample by patient, and then we identify which are the allele of a patient. So we identify all of the possible alleles within one patient. So when we align against this. Specifically, if you're looking at the SFS plot, which I forgot where I put at the end, I think. If you're looking at the SFS plot, the sort of where you expect to find allele variant, you expect them to pop up as like for specific gene to pop up as like spike at the very end or at like 50% of this sort of curve. So in our case, we checked for the different gene and we don't see that. So we expect that allelic variant don't pollute the result. But I don't think it would change any of this sort of argument because we're comparing expressed versus binder. And so that's where we see the difference. And I think it wouldn't imply. But yeah, I agree that for general, when looking at SFS, it's a problem.
Great. Thank you. And then Stan, I will pass it back to you.
Yeah, I actually have a bunch of questions regarding the tight-seq because I think you use tight-seq in a couple of research papers, and it looks like it's very, very nice method for measuring the affinity. But how? And I see that some people in the industry starting to use this approach because it allows to test on the like high throughput. Yeah, but my question is how accurate it is, and whether there are some caveats like in applying this method. What kind of depths you need to do, like what to pay attention to make it precise?
Yeah, I think in terms of accuracy, we are like fairly similar to what you would obtain if you just do titration for individual yeast. Do I have this right? But yeah. Yeah, we have a sort of plot. So we tested against bilinear interferometry. We tested against standard titration methods, or that is published. Affinity seems to be the accuracy seems to be fairly good. But we do need to sequence much more than if you are doing just a simple screen. That's why in this project we use barcode to be able to just sequence the specific barcode and not the full antibodies, which greatly limited the cost of sequencing. And I think sequencing at the end of the day was fairly cheap, given the amount of data that we got out of it, probably around. I didn't want to say something wrong, so I'm just not going to say it, but like it's way below 5,000. Of course, yeast are not human cells. They have different biotinylation, so they behave differently. I don't think that's always an issue that we need to think about. That's always a potential problem. I don't think this is the main issue. I think something that I think is more important is, for example, the fact that all the measurements that are done in the industry and in academia are done at equilibrium, and that actually antibodies are basically never at equilibrium when they interact with cells. They get pulled, they get everything. So I think that modifies a lot of the antibody react. So I think that compared to that, the fact that we're looking at like, slightly different glycosylation level and things like this, and expression issue is not too bad of a problem. But yeah, it's good to keep in mind that we're working with yeast, working with SCAV, not real antibodies.
Yeah. And in your another question. So in your CFV construction, so you use the combinatorial combinatorial approach. But if you know, there are some models that kind of predict pairing, the natural pairing of heavy and light. So there is a data set generated by 10x Genomics, and then there is a immune match, a machine learning model trained on that data set to actually predict. Do you think I mean, just maybe, did you test your like pairs and the CFEs that work nicely against this predicted score?
So no, I'm aware of the 10x dataset, but not of the prediction methods. So yeah, definitely, I probably need to do that. But yeah, good to know.
Yeah, I'm asking because like we have a data set of single cells in an antibody discovery campaign. It's published data set where we looked on the correlation between like those antibodies that were selected by the immune system naturally and have a high affinity against this machine learning model that predicts this pairing score. And there were some nice correlations actually. So like there is something maybe interesting. Going away from this, another important question that I think many people will listen when you mentioned this were really curious. You mentioned AlphaFold performance. Yeah, can you elaborate? How did you use AlphaFold? Well, technically, so how did you use it? Like which tool to predict the affinity, and whether do you think that's a fundamental limitation of like today structure modeling approaches, or is just this data set and this AlphaFold tool? And let's say if you use Immune Builder or something else, it will work better.
Yeah. So we use AlphaFold so we use the 10 for each of our antibody. So this is a lot of antibody, and we didn't have that much compute time. So for each antibody, we use 10 seed using the last version of AlphaFold, like 8 months ago, I think.
Sorry, the last
And
AlphaFold
we took
3,
the
you mean?
best of the EPTN.
AlphaFold 3 or AlphaFold
Wait,
2?
no. This was the V2 because we needed to run it on our server. So we didn't. So of AlphaFold V2, 10 seeds per antibody, and we took the best of the IPGN. For the internal representation, so you can basically access the intermediate layer, layer representation of AlphaFold, and we used that, put that in a UMAP, and did very basic things to get this sort of UMAP-shaped internal representation. So it was not particularly, yeah, we followed the manual basically on all of this.
But do you think the fact that AlphaFold is not doing very well here?
Oh, yeah, sorry. The other part of it.
Yeah. So do you think with
so
other?
I think that the fact that it kind of succeed in grouping antibodies that are similar together mean that it's probably able at some point of measuring affinity and getting some idea of affinity correctly. Okay, my point of view, which is very biased, is that this is because we don't have enough data, and so we need to get more data into the machine, and the machine will get us better results. But honestly, my knowledge of this type of model is fairly limited at the end of the day, and I have no very clear idea on like what could work and what could be done to improve them in this type of thing. And maybe it's possible to actually improve them directly.
Yeah, well, thank you very much, Thomas. My last question actually is, yeah, sorry, Monica, I have one more question, the last one, because again, it's very interesting for our audience. So there is affinity, but then in the industry or like in academia, when we do antibody discovery, there is also developability. If we cannot manufacture the antibody, it may be useless. So the question here, like, on the one side, that's how I think immune system does not care whether we can manufacture antibody or not. But on the other side, that's my question. Do you think we can somehow use natural immune system to, I don't know, help us find antibodies that not only like have high affinity, but also have better developability properties downstream?
Well, it's that's a big subject. I think there's evidence that part of the selection during affinity maturation is done not on affinity, but also on how easy the antibody can express, right? So if we can understand affinity maturation enough and understand this system enough, we may be able to get antibodies that are not just better binder, but better, easier to express. But then the expression in industry is very different from expression in the body. So I think the answer is like, I don't know. I mean, I can tell you if the antibody express well on yeast, but that's very much the limitation of my method.
True. Thank you very much, Thomas.
Thank you so much, Shoma. So I'm just going to share our last